Dr. Steve Winitsky is a medical officer in the Office of Cellular, Tissue, and Gene Therapies in the FDA's Center for Biologics Evaluation and Research. As a medical officer on the general medicine team in the Division of Clinical Evaluation and Pharmacology Toxicology, he conducts primary clinical reviews of investigational new drug and investigational device exemption applications for biologics, devices, or combination products intended for non-oncologic indications. His topic today, the regulatory pathway of cellular therapy. So please help me welcome Dr. Winitsky. Thanks for being with us today.
Thank you. Is it the clicker? Okay. Okay, great. Hello. So today I'm going to speak about the regulatory pathway of cellular therapy. Cellular therapy you're going to hear a lot about at this meeting. You've already heard a lot. There was a very nice talk by Dr. DeBaria. There are a lot of aspects of cellular therapy that are very attractive. It has great potential for a therapeutic for cardiovascular disease. And what I'd like to do is give you some idea of the steps that are important in the regulatory pathway when you're moving forward. It's a relatively young field, and a lot of the trials are phase 1 trials. So I'll focus on phase 1 trials and submitting IND. IDE is similar pathway for devices. There are some differences, but I'll be focusing more on the IND pathway. As a brief overview, we'll talk about clinical development programs and some of the important considerations and where phase 1 falls into that development plan. We'll speak on some basics of IND, investigational new drug applications, considerations that are specific to cellular therapy trials, or at least are important, not specific, and what we term pre-IND interaction. So the interaction between sponsors and who are submitting these trials to FDA prior to submission of a formal IND application, and then some of the important components of an IND application that you should give consideration to when you're putting the packet together. The clinical development program, as we all probably know, involves a number of phases, preclinical, phase 1, 2, and 3, and that's all to try to get a product approved and on the market. Currently, there are no approved cellular therapies for cardiovascular disease in the United States, but there are a number of trials that are, a number of programs that are advancing in phases, and some exciting developments, of course. Phase 1 trials, which we'll be talking primarily about today, are usually small studies, and these are focused primarily on safety. And I'll discuss some of the differences between phase 1 trials of cellular therapy and additional drug pathway. These trials, one difference just early on I'll say is that they usually do also assess bioactivity instead of being pure safety studies. For those of you in the drug realm, those are the studies that you're used to for phase 1. For phase 2, we're talking about expanded safety and efficacy studies. An important consideration is that you really need to optimize study procedures prior to getting to phase 3. These are optimally done in phase 2. Further dose exploration goes on at this point. Phase 3, of course, the studies need to be designed to provide substantial evidence of effectiveness and also a further indication of safety. Phase 3 studies, the reason I'm bringing up phase 3, really talking about phase 1, is just to give some idea that you should be focused on getting an overview of what you'll need to collect in the different phases to be able to try to get a product approved. One of the best guidances that I think we have is guidance for industry providing clinical evidence of effectiveness for human drugs and biologics. And there's a link at the this this discusses the quantity and quality of evidence that you need to demonstrate effectiveness. And if you don't think about this, at least give some thought initially, you can be surprised as you think you're progressing in the process. So it's always a good idea to consider this up front. Think what you need to collect in the different phases, and then you can analyze whether your trial is designed to meet those standards that you need to. So an investigational application, which we call an IND, that's required to conduct a clinical trial of an approved drug or an approved product for a new indication or in a new patient population. The regulations you can look up if you need to in 21 CFR 312. FDA has 30 days to review the new IND application and to make sure that research subjects subjected to unreasonable risk. So some of the differences between cell therapy and drug trials. Cell therapy trials often enroll patients who have the cardiac disease in question, and drug trials often enroll normal volunteers. As we talked about, bioactivity is important to assess in phase 1 in cellular therapy trials. And control groups that undergo invasive study procedures may enhance the interpretability of the results of the cell therapy trials, but there may also be concerns such as ethical concerns, risk-benefit considerations. So those need to be taken into account. Just one thing to point out, what we think would be an optimal trial design is not always feasible, but we strive to get a trial design that really can provide the best evidence of safety and activity. And then that's taken into, you have to take that into account in the context of the overall clinical development program. So the duration of follow-up for efficacy and safety may be longer for cell therapy trials, and that's due to a number of reasons. The time it takes for that product to actually work, and also the potential for having adverse events. For cell therapy trials, especially that involve autologous products, the same number of a pure population of well-characterized cells is rarely administered to individual subjects, which can complicate the interpretation of bioactivity. And when you heard Dr. Perrin, for those of you who attended the morning session on the FOCUS trial, you could see there was some pretty big heterogeneity in the cells that were administered, and those potentially can have effects on outcome and may complicate interpretation of results, especially for small early phase trials. Now, this is a guidance that's specifically designed for sponsors who are studying cell therapy. Here's the link on the bottom. This provides some general guidance on regulatory issues and trial design. And I'm bringing that up because it's a good place for starters when you're getting some information about what these involve, some of the important elements. So cell products are defined as biologics and regulated by CBER. The products that we regulate at CBER, there are other definitions for biologic, but in general that's cell therapy products and gene therapy products, and things like monoclonal antibodies are not regulated by CBER. So the intravascular catheter is one major difference between drugs and biologics. So when we're talking about cardiac trials, as Dr. DeMaria was mentioning, a lot of times you use a device to deliver the cell therapy, and that we call a combination product because you're usually talking about a dedicated delivery system. So you should be aware of the implications of that, and in this guidance, it talks about how we regulate these kind of products. Now, having said that, all the guidance for cellular therapy, I mentioned as a good starting point, we recommend very strongly pre-IND interaction with the agency. So this is prior to actual submission of a formal IND application. So the guidance document cannot, of course, take care of every situation for every type of product and development program. So the pre-IND program allows for early communication between sponsors and the agency so that you get some guidance and feedback on things that are really going to be important for IND application. As I said, we strongly encourage this, and we can't emphasize enough that an open dialogue between sponsors and the FDA is really critical and gives you the best chance for conducting an efficient development program. Some of the benefits are that you get feedback from CMC, which are the branch that evaluates the manufacturing process, preclinical branch, clinical branch, and statistical branch, and you can request any or all of those branches. You do need to ask specific questions, and you will get those questions answered either in a face-to-face pre-IND meeting or in a teleconference with the agency. So the pre-IND package should contain an outline of the preclinical program, CMC, and the proposed trial. But for us to be able to provide helpful comments, we really need to see some more detailed data than just a general outline. But we can certainly apprise you of the major considerations. So some of the contents of an IND application, we'll go over some of the elements. A commonly overlooked area is the rationale, and this we actually focus on quite a bit when we're reviewing an IND application. We'd like to see why you think that the method of delivery, why this cell in particular, why the study population, why you think that based on your preclinical data and other clinical data that's available, why you're deciding to move forward with studying this in humans. The clinical data is listed, trials using the same agent, trials of related products, and the same or related delivery devices. Some farm tox information that's important are all in vitro testing that's been performed, farm tox study conducted in animals. Some of the data that's important is listed, bioactivity data from this. This helps us evaluate safety and potential activity in humans. CMC information, it's the related to the manufacture of the product and also gives some idea of identification characteristics of those cells, quality and purity. Bench testing is especially important for the catheter, issues like biocompatibility. One thing that can be pretty helpful is cross-referencing of files, of regulatory files. So for instance, a master file, let's say for a device, or other INDs. So you'll need permission from the sponsor of those INDs or files to be able to reference them, that is cross-reference them in your IND application. So part of the benefit is that it avoids repetitive submission of the same information, can often preclude the need to repeat studies that have already been done previously. However, we're not allowed to communicate with you the contents of what is in that IND or master file. So specific deficiencies in those files that you may not know about, because of that information contained in those files might not satisfy the issues that you think it's satisfying. So part of the protocol elements that are important, I'll just list some of them. The endpoints, an important consideration is that the endpoints are not necessarily the same for all phases of development. And this may sound obvious, but you really need to focus on why you're doing that phase trial, what information you're collecting, and the feasibility. So in general, phase 1 studies use multiple endpoints, and the reason is that it's important to collect as much relevant data as possible. However, improper analysis from the multiple endpoints can cloud the interpretation of those results and may lead to misrepresentation of product's activity. The primary efficacy endpoint, importantly for a phase 3 trial, must reflect a clinically meaningful effect of the product. And that's just something to keep in mind. So study population considerations. Early phase trials often enroll a very narrow subgroup of the total population that has a certain disease. This is done for risk-benefit considerations and also issues related to interpreting safety and activity assessments from that trial. And it's important that later phase studies strive to enroll a more general population if you want to address that issue of generalizability. And there are a number of trade-offs, and during the pre-IND process, we can certainly discuss the issues if you have questions about that. So some more considerations for the study population. It's important for biologics and cell therapy trials that study subjects must receive the standard of care therapy, and if feasible, try to exclude vulnerable populations. And while we're on that topic, for those of you who are studying things like congenital heart disease, planning on studies in pediatric patients without adult data, first for the studies, 21 CFR 50 and the subparts listed list some of the considerations that are critical for studies. And for instance, studies involving greater than minimal risk, studies involving greater than minimal risk but presenting prospect of direct benefit to individual subjects. Determine which category your trial fits into, and read these passages carefully so that you know what standard you'd be held to. Enrollment in the phase 1 trials, usually there's a staggered intra-cohort and inter-cohort administration of the investigational agent to subjects. This is done as a safety feature of trials. So if you see a safety signal arising, enrollment of subsequent subjects can be discontinued, and that can be done pending investigation of the safety of what's been administered already. Controls and blinding are often closely linked. The choice of one will affect the other. You have a number of options for controls: no control, a standard of care comparator, a sham procedure, a placebo control. And it's important to remember at the heart of all FDA assessments is a risk-benefit assessment. So in particular, the risk of the control procedure, the actual data that you'll be collecting, and whether the benefit of collecting that data outweighs the risk, especially for early phase trials. And blinding, open label, no blinding, single blinding, double blinding may be very important to assess activity. Safety monitoring plan there are a number of considerations. It should take into account existing data, preclinical or clinical data, and any unique characteristics of the product. Stopping rules should be strongly considered. These are rules that if a certain number of events are seen, these stopping rules are triggered and the trial is temporarily halted while a safety assessment can be conducted. DSMB, a Data Safety Monitoring Board, can be helpful. Duration of monitoring for efficacy and safety is dependent on a number of factors, but the assessment of long-term activity is fundamental to the ultimate determination of the risk-benefit ratio. And appropriate follow-up to monitor for adverse events is, of course, warranted. Some of the important assessments for these cardiac trials that use cell therapy: mortality, cardiac events, abnormalities in markers, which would be, although not an event in itself necessarily, or sometimes can be a biomarker for cardiac injury with catheters, actual damage to the heart, perforation, and pericardial effusion. And a lot of these products may be allogeneic. You may generate an immune response, so to study the cellular and humoral effects of the product when they're administered. Phase 1 analyses, statistical analyses are generally exploratory and are not powered to demonstrate significant differences in outcome, but can give you some preliminary evidence of efficacy. And it's important to consider pre-specification of the analysis, as that improves the interpretation of the data. And this is a website, and here's the link. This lists some webinars that you can just click on and look at all the different topics, so you can get some idea of what you'll need to consider in your IND. At the moment, it's preclinical and CMC topics, and the clinical talks will be added later in the year. And here's my contact information.
All right, how about a nice thank you for Dr. Winitsky?